Trees have existed for more than 100 million years on our planet. Throughout this time, they have had to adapt to countless environmental and climate changes. Adaptations occur through genetic and evolutionary changes over generations, during fertilization and the dispersal of the produced seeds. The most recent changes of our trees' habitats occurred in Europe after the last ice age, which lasted up until around 15,000 years ago. Until this time, the Alps were mostly covered by glaciers, and many of the ice-free areas were covered by a tree-less steppe, similar to today's tundra. Small tree populations could only survive in the Danube basin and in the eastern alpine region, which was partly free from ice. Due to repeated shift from warm to cold periods within the last 2.6 million years, the diversity of tree species in Europe decreased significantly. Only 29% of all woody plant genera, which were present in the Tertiary period, have survived the manifold climatic changes of the past. During the last glacial periods, most tree types were growing in retreat areas, so-called refugia, in southern Europe. With the end of the Ice Age, since about 15,000 years ago, temperatures increased and the different species started to spread north. Based on discoveries of fossil wood and pollen, the migration of tree species out of its refugia could be reconstructed. Sessile oak began its journey to the north approximately 14,000 years ago, and it lasted 8,000 years before it had spread all the way to Scandinavia. The patterns of the spread after the Ice Age are still existent in the genetic heritage of the tree species today. The long regional isolation of tree populations in the refugial areas resulted in different genotypes and genetic structures due to genetic drift and local selection. Even today, these genotypes tell us where our trees originate. Though north of the Alpine range, many migration lines met and intermixed with each other. But how can we explain this migration of trees? Because aren't trees rooted at one site over decades and even centuries? The solution can be found in the seeds, which can travel great distances in a short time. From the reconstructed migration path of sessile oak, we can estimate a dispersal speed of between 150 and 500 meters annually. These long distances are unexpected for a tree species whose seeds, the acorns, are relatively heavy and mostly just fall and land few meters away from the original tree. The high speed of spreading can only be explained through rare long-distance seed dispersal events. For example, the jay has been found to transport acorns over several hundred meters or even kilometers. Acorns can also be transported along streams and rivers. Other tree species, especially the native conifers, have small and light seeds that can easily be carried by the wind. Successful dispersal of spruce seeds were found to occur over many hundred meters, up to a kilometer, and with the right wind conditions, even dispersal across several kilometers is not unlikely. Besides seed dispersal, trees, or rather their genes, also spread by means of pollen flow over large distances, and thus pass local adaptations to climate and location to other forest stands. Particularly pollen dispersal by wind can quickly extend over several dozens to 100 kilometers. Both dispersal processes, seed dispersal and pollen-mediated gene flow, play an important role for the adaptation of forests to climate change. However, pollen and seed dispersal are multidirectional processes and do not, for example, blow locally adapted pollen from southern Europe to the north. Instead, the direction of these processes is mostly random in terms of climatic zones, and can therefore provide only limited help for adapting our forests to the future climate. For this, a directed genetic adaptation to future environmental conditions would be required. For a long time, scientists believed that the genetic structure of tree populations and any other organism could adapt only very slowly, by means of natural selection. The paradigm since Charles Darwin was that the phenotype, meaning the appearance of an individual, is affected by its genotype, and the manifestation of the phenotype by the environment. Here, genotype refers to the genetic information stored in the DNA. In this concept, only the genotype could be passed on to future generations, but the phenotype, which is a result of the interaction with the environment, could not. This paradigm was seen as a fundamental concept of understanding evolution until the end of the 20th century. However, in the 1980s, Norwegian forest tree breeders started to challenge and modify this view of evolution. Seed production at the northern tree line is low due to extreme weather conditions. However, for reforestation of northern latitudes, tree breeders aimed at utilizing seed material well adapted to the harsh environmental conditions. This is why they transferred northern genotypes with genetic characteristics from the north to the south. That is a more suitable climate for seed production. Here, the established seed orchards are expected to produce seed material for the northern growing regions. With the establishment of seed orchards with northern genotypes in southern Norway, the breeders expected to produce seeds adapted to northern environments. And these seedlings were thought to possess the same attributes as the plants grown from seeds fertilized in the north. However, a part of the desired phenotypic traits, such as bud flushing in spring, bud set in autumn, growth rate, but also frost resistance in autumn, were lost. In the following years, the Norwegian researchers worked hard to understand how seeds with genotypes from the north, but fertilized and matured in the south, could have lost their northern characteristics, their adaptation to the cold. They realized that the environmental conditions during fertilization and seed maturation affected the characteristics that the plant would later have. This implies that the trees are affected by environmental factors during seed production, and the trees remember these environmental conditions for a long time. Today we know that environmental effects can affect gene expression, cell characteristics, and complete phenotypes by means of epigenetic modification, and that these environmental effects can also be inherited to the next generation. The basic molecular mechanisms of epigenetics mainly include histone modification regulating, for example, the organization and packaging of genetic material, the function of the ribonucleic acids, RNA, for regulating gene expression, and a methylation of the nucleobase cytosine, which is one of the 4 primary units of the genetic code. Such a methylation of cytosine prevents the RNA polymerase to read and transcribe the respective gene on the template strand, to which the methyl group is attached, and thus there is no translation into a functional protein, for example an enzyme, through which different genetic functions can be switched on and off, and through which a specific phenotype can be expressed. Scientific studies on epigenetics are currently taking place mainly on model plants with short generation times, such as the thale cress. If the manifestation of a phenotypic trait is affected by environmental conditions, and if this effect is inherited by the next generation, it can be investigated mainly along changes from one generation to the next. Such studies demonstrated that the epigenotype can be stable over many generations, but it may be altered under certain environmental conditions. For Norway spruce, the temperatures at the time of embryonic development, the time of seed maturation, and the environmental conditions during seed germination were found to be sensitive life stages. Thus, higher temperatures in May and June, shortly after Norway's spruce flowering, were found to result in seedlings that have a longer growing period in autumn and set winter buds relatively late in the season. At locations with early frosts in late summer or fall, trees with such seeds will be more susceptible to frost events. On the other hand, such seedlings utilize the vegetation period more effectively and show a better overall growth. An open question is if epigenetic adaptations might also support trees to withstand the expected climate extremes, as for example the increasing frequency of drought periods. The project Adaptation at the Austrian Research Center for Forests (BFW) investigated whether our forest trees are prepared for the ongoing climate change and whether changing climate conditions during reproduction are affecting the performance of the next tree generation via epigenetic modifications. A major objective of Adaptation, which was supported by the Climate and Energy Fund, was to analyze the influence of natural weather fluctuations at the time of tree flowering, fertilization, and seed maturation on adaptive variation under warm and dry growing conditions for young trees. To test whether adaptations to a warmer climate can be acquired by environmental conditions during reproduction, forest seeds from Norway spruce, European larch, and Scots pine, available from different maturation years, were tested within a 2-year seedling study. The selection of maturation years considered the weather conditions during tree flowering and seed maturation. Loosely speaking, these can be categorized into cool, moist, and warm, dry maturation years. In addition to that, the grown seedlings were tested under 2 environmental conditions. One treatment received normal precipitation conditions, while for the other one, precipitation was reduced by 50%. After every vegetation period, some plants were removed from the experiment and various measurements were taken. Moreover, drought stress experiments in a greenhouse were carried out, along with the analysis of frost resistance in winter and early spring. The project result confirms the central hypothesis and show that climate conditions during reproduction and seed maturation have a significant effect on the phenotypic characteristics and the vitality of young trees. The experiment revealed the largest effects for parameters of productivity, such as seedling weight or height. Seeds maturated within warm, dry years tended to be smaller and lighter for all 3 species. In particular, these seedlings showed lower phenotypic plasticity than seedlings grown from seeds matured in cold, moist years. But the climate conditions also affected characteristics that are important for survival under water-limited conditions, such as the proportion of the root length to the total seedling size. Here, significant differences between the 2 precipitation treatments were found. Seedlings in the 100% type were bigger and heavier and had a higher root-to-shoot ratio. All results clearly demonstrated the effects of weather conditions during tree flowering, fertilization, and seed maturation on phenotypic and physiological characteristics of young trees, and suggested that seedlings grown from seeds matured within warm, dry years have a pre-adaptation to potential drought. However, these seedlings also seem to have a lower productivity and plasticity, and therefore cannot be generally recommended. Instead, the project results prove that further research is highly needed to understand the effect of environmental and climate factors on the epigenetic control of seedling and tree characteristics, until specific conclusions for practitioners can be drawn. Another critical time period for the acquirement of specific phenotypic traits is the time of seed germination and early seedling growth, as demonstrated in recent studies on young Norway spruce and European larch. Slovak researchers compared plants of these species which were germinated and grown in 2 different nurseries at different altitudes.
Und sie haben festgestellt,
Our colleagues in Slovakia found that seedlings which germinated at a higher altitude had different characteristics than those of the same genotype which germinated at a lower altitude. For example, seedlings germinated in the higher altitude nursery had a lower temperature requirement for needle flush than the seedlings germinated at a lower altitude. This lower temperature requirement is a clear adaptation to the colder climate of the higher altitude nursery. A full understanding of how environmental factors affect the development of young plants and their seasonal growth patterns is not possible yet. However, these results demonstrate that trees and their genes interact with prevailing environmental conditions. A better understanding of this interaction is highly needed in order to consider it during seed production and reforestation for climate change conditions. Above all, we need to answer an important question. Is the ability of our trees to adapt naturally by epigenetic changes, the natural migration speed of our trees, high enough to adjust to the expected temperature change of 2 to 4 degrees Celsius caused by human climate change in less than 100 years? Climate envelope and species distribution models can be used to model the shift of the optimal growing regions for different tree species. Norway spruce, for example, might easily migrate into higher elevations, because here the spatial distances are small. However, the horizontal migration northwards would need to cover large distances, and this will not be possible for many tree populations without human help. Norway spruce and many other tree species are expected to shift their distribution range, and these shifts are needed if the species is going to survive in the future. In the past 10 years, researchers around the world investigated how trees, but also other plants or animal species, might be transferred into other regions and new habitats, which can act as climate refugia in order to save their genetic diversity, and maybe the species in general. This research and nature conservation concept is referred to as assisted migration and deals with migration assisted by human intervention. With this concept, we would not only support the survival of trees and their valuable genetic material, but we could also provide forest managers with the appropriate valuable seed material to manage our forest ecosystems sustainably also in the future.